All right, we're getting to 1.4b gene expression and mutation. And after I close this antivirus pop-up, let's continue. This is the best source you'll ever get on the subject. Mutations. This subject will be going to talk about a lot about mutations. Mutations are just changes in the DNA sequence. And your base DNA sequence are the nucleotides A, T, C, G. And here is a picture of the original sequence. But you can have mutations that do weird things. In this example, the T turn into a C, and that would be a point mutation. We'll learn more about this later on, so let's move on. Know the difference. Last video, we talked about the two ways to get cancer. There's either a mutation, which can happen due to changing the DNA sequence, and there is epigenetic changes. And this happens, epigenetic changes the gene expression without altering the DNA sequence. The learning intention 1.4.5. Mutations in genes and chromosomes can result from errors in DNA replication or cell division, or from damage by physical or chemical factors in the environment. Let's move on. Mutations, when do they happen? Well, it usually happens during the S phase of the cell cycle, and that is the phase where DNA is synthesized, S for DNA synthesis. And during that phase, the DNA is unwound, it is exposed because, to replicate DNA, you have to unwind it first. You have to unwind the two strands, so the polymerase can add spare nucleotides to the exposed strands. But this is also giving, UV rays, for example, a chance to um, change these vulnerable DNA sequence. Remember, the S phase of the cell cycle is at the time where the DNA sequence are most vulnerable to being changed. And during this time, UV rays, if you go under the sun without putting on sunscreen, UV rays will probably damage your DNA a little bit. And this is also the S phase of the cell cycle is also the phase where DNA is getting copied. So if you messed up your DNA during that phase, more of the DNA with errors will be copied. And you don't want that because you want proper protein, you want good protein, you want good protein made from good DNA. Bad DNA will just produce cancer. And mutations also happen during cell division, such as mitosis or meiosis, and it happens when they divide unevenly. And next slide, mutation types. Here we're learning about point mutations. So point mutation is just mutation to one letter in the DNA sequence. There are 4 point mutations that may happen during DNA replication. There is deletion, just literally take one out and there's nothing to fill it in, which is not good. I mean, what good can happen when you have one less DNA letter? And there is insertion, which is literally just put one more, just somehow put one more DNA base in the middle of anywhere. So you could have just normal DNA and you put a C in there, but all these DNA sequences coming after that one would be messed up because they have all been pushed back. And since the order that these DNA coming are so important, if you insert one, you can significantly change the structure of that protein that this DNA codes for, which is not good. And there is substitution, which is relatively not very harmful because substitution just takes out a letter and substituted the letter back in. There is no insertion, there is no deletion, and sometimes there is no change at all because that it can cause a silent mutation, which is not observable. And final, we have inversion. It's inversion of adjacent nucleotides. In this example, AT turns to TA. And here we're learning about the effects. The effects of DNA mutation. There is missense, which is straightforward. It changes the protein that is being produced. There is silent mutation. And these silent mutation happens because we know that the the DNA nucleotide sequence can produce more than let me rephrase myself. More than one DNA nucleotide sequence code for the same protein primary structure. Let me say it again. The same protein primary structure can have different sequences of DNA nucleotide. So here you can have GLY, and the DNA nucleotide for that could be GGT or GGA. Either way, it works. And if you replace the T up here with the A down here, nothing will happen because the same protein is getting produced. And this is a really interesting thing that the DNA has. Like if you just mess up a little bit, there is a slight chance that the mutation might do nothing, and that decreases the chance of a serious mutation. And nonsense, this is really bad. This is one of the worst because nonsense just stops the protein, the polypeptide chain that produces protein. Before you had CYS, but after you replace the T with the A, you get a stop protein. This would be normal if it was placed in the right position, but down here, you're just cutting off a chain in the middle of protein production, which is not good. That's nonsense. And frame shift. This is bad. This is bad. Frame shift makes massive changes. You see how it's in red font? Big impact. And this happens when you insert a letter into the DNA nucleotide sequence. Because the letter is inserted, all the letters coming after this one is going to move back by one space. That may seem like nothing, but essentially it changes all the proteins that is being produced. You see all the proteins up here and down here are totally different. Totally different. And there is another mutation type before we learn about what is it? DNA mutation types. All right, down here, chromosomal mutations. And this happens in the structure or number of chromosomes. So before, over here, we're learning about the mutation that happens with your DNA nucleotide sequence. But here, we're talking about the chromosomes on a larger scale now. And this happens during cell division, mitosis. There's translocation. Genes are exchanged between non-homologous chromosomes. Happens during meiosis. Duplication, a gene may be replicated more than one time, creating extra copies of a gene on a chromosome. So part of the chromosome may just be copied more than one time, and the new chromosome is essentially lengthened, and it contains two parts of the same genes. Deletion is just deleting part of the gene from a chromosome, and as a result, it gets shorter. Inversion is when a section of chromosome breaks off and the broken segment is reversed and inserted back into the chromosome. So this part is just upside down. The rate of mutation. Under intention 1.4.6 mutation rate can be increased by ionizing radiation, mutagenic chemicals, high temperature, and viruses. So stay away from those four. Factors increasing mutation types. There's radiation, could be UV rays, could be any other ray, X-ray, gamma rays that will kill you. And UV rays may break sugar phosphate backbone, and they may cause errors in base pairing. And this is how you mess up your DNA. So one of the stupid things that people do is one of these tanning beds. Because what it does to you is it just shines UV rays at you to get yourself burnt, to get yourself tanned. Although you may look quite good after, this what this literally does is it applies UV ray to your body, to your skin cells, and essentially kills some of the skin cells and may affect your DNA. And what this could essentially leads to is skin cancer. So that's why if you're paying to get tanned, probably don't just go to the beach, get some vitamin D from the sun or whatever vitamin you get from the sun. You know, the sunlight is more healthier than just UV rays. And radiation, and they can break up strands of DNA because it's stronger than the UV ray and it causes more serious damages. And another factor that increases mutation rate is chemicals. There's a word for it called carcinogenic, means having the potential to cause cancer. I would say a cigarette is carcinogenic. Some drugs could be carcinogenic. UV ray could be carcinogenic. But anyways, let's move on. Asbestos. It includes mutations in special genes that help with tumor suppression. Now, this is just example. Yeah, a chemical that could increase the mutation rate. And what this chemical called asbestos does, it induces the mutations in special genes that help with tumor suppression. So what it will do to your body is um, induces mutation. So you don't want to mess up any genes that is responsible for tumor suppression. And if you mess it up, your body could replicate cells at a undesirable rate, at a abnormal rate, faster. That's how tumors can form. They're just abnormal cell growth. Factors increasing mutation rate. Viruses, they can cause mutations by putting viral DNA into the human DNA. And the human DNA can also be called genome. They may insert oncogene into genome. What oncogene does is exactly the opposite of tumor suppressing gene. Tumor suppressing gene slows down the process of cell division, whereas oncogene just speeds it up. And there's also high temperature. High temperature can break up chromosomes. It can denature enzymes that are crucial for DNA functions. Now, this is the part that is really important, somatic versus germinal mutations. Somatic cells are all the body cells except reproductive organs. And so somatic cells could be your skin cell, your liver cell, your cheek cell, any cell but your cells of reproductive organs. So a sperm or an egg would be a germinal cell, it would not be somatic cell. And any mutation in your somatic cell are localized. They are in a specific area and they're not inherited by offspring. Well, for example, if you get skin cancer and it's only affecting the somatic cells, it will only stay on your skin, right? It's often localized, means it's probably not going to transfer. And when you have a child, is the child going to have higher rates of skin cancer than other children? No, because if the cancer is only affecting the somatic cells, that risk of having cancer is not going to be passed down to the offspring, which is good, you know. But what you might be scared of is germinal cells because germinal cells are in reproductive organs. And mutations in these cells can be passed down to to your offspring. When the germ cell became gametes, and they are present in every cell of the offspring that arises from these gametes. Means if the cancer affects one of the germinal cells, the offspring is going to have a higher rate of getting cancer. And these are a list of keywords to remember. Let's go over them. Epigenetics, genome, epigenome. Genome is the complete set of genes in a cell. I'm not typing twice, which is not good, in a cell or organism. Epigenome, the unique pattern of methylation and acetylation that affects how the genomes are expressed. Genome and epigenome both express the both affect how you look. They both affect your body function, your behavior, and a lot more other things. But essentially, genome, simply put, is all the genes, all the ATCGs in your body. And epigenome is the epigenetic factors, methylation, acetylation, anything that affects gene expression without changing the base DNA sequence. Next, we're moving down to DNA methylation. Involves the silencing of a gene through the addition of methyl groups to cytosine nucleotides in the DNA. Methylation silences a gene by inhibiting the function of RNA polymerase during transcription. Acetylation is a process in which negatively charged acetyl groups are added that weaken attractive forces between the negatively charged DNA and the positively charged histones, forming loosely packed euchromatin. Histones are proteins that act as spools which DNA winds around. Nucleosome is a complex consisting of a length of DNA around the core of histones. And there's euchromatin, has negative acetyl groups attached as condensed. Heterochromatin is just the same thing without that negative acetyl group attached to the DNA and histone, and it is more condensed. Such transcription cannot happen. Transcription factors. Activator is a protein that increases gene expression of a gene or set of genes. Repressor is a protein that inhibits the expression of genes. The DNA binding repressor blocks the attachment of RNA polymerase to the promoter, thus preventing transcription of genes into messenger RNA. Translation factor. Translation factors function to coordinate and catalyze the synthesis of polypeptides from mRNA templates by templates. That doesn't make sense. Huh? Yeah. So transcription factor takes care of transcription side and translation factors takes care of translation side. Promoter sequence is a region of DNA sequences that define where the transcription of a gene by RNA polymerase starts, and the promoter sequence is actually not uh, transcribed. Uncoding RNA is a functional RNA molecule that is transcribed from the DNA but not translated into proteins. They don't code for the protein, thus they're not translated into proteins. Tumor suppressor are normal genes that slow down cell division, and proto-oncogene does the exact opposite. They're normal genes that provide signals that lead to cell division. And thank you, that is the last slide. Have a good day. Enjoy biology.